everyone, this is Shraddha and you are watching Knowledge Center. In this lecture, I will tell you about the active site, enzyme active site. So, what is enzyme and what is function of the enzyme, property of the enzyme and classification of the enzyme. I told you in the before lecture. In this lecture, I will told about the active site. So, what is the definition, then how active site attach with the substrate, then features of the active site, mechanism of enzyme catalysis and binding substrate theory. There are two important theory, lock and key theory and induce fit hypothesis. Another three theories are also four theories. Okay, so let's get started from the active site. So what is the active site? So the site in which a substrate molecule get bind, it is called the active site. Means this and this is the active site. Total this structure is called the active site where the substrate get bind. Okay, so the size of the molecule, enzyme molecule is a greater than the substance. Substance is lower size, lesser size from compared to the enzyme. And this that believe that the only small portion of the enzyme molecule comes in the contact with the substrate, and this region is called the active site where the substrate get be attached. So the enzyme molecule made with the protein, and protein is made with the amino acid. So enzyme have amino acid, so there this active site contained amino acid residue exposed, some residue exposed where the substrate molecule get bind and interact to form a enzyme substrate complex. Okay, and how they bind? So they bind through the hydrogen bond and through the ionic bond and the covalent bond. Okay, so ionic bond is a donate electron, matlab one atom donate electron to the another atom, that is ionic bond. Means if the type of chemical bonding that involve the electromagnetic attraction between the opposite charged ion. Okay, so this is ionic bond. Hydrogen bond means it is a electrostatic force of attraction between the hydrogen atoms of the two atom. And covalent bond, it is a chemical bond that involve the sharing of the electron pair between the atom. Means what is the difference between ionic and covalent? Ionic have a donate electron to another, and covalent that the share the electron. So the enzyme get attached with the substrate of any bonding and make a complex, then complex convert to the product, and then product dispatch with the enzyme, and enzyme get back their original position. Now how active site attach with the substrate, I told you. Now feature of the active site. What is the feature? So unique shape. The enzyme active site have a unique shape. So unique shape of substrate can attached in this active site. Okay, and a small pocket, form a small pocket. So it is form a small pocket in the enzyme get formed one or more region of the polypeptides. Okay, and take place a small portion. And next one is a proper region. So the active site have a proper region where the substrate bind and make a substrate complex, and the complex intermediate between the substrate and the product. Okay, and the complex then decompose, and product found the product, and when then dissociate from the enzyme, and enzyme get back their original position. Next one is the specificity. So the specificity of the substrate binding depends on the arrangement of the atom. Arrangement of the atom means that this enzyme substrate make an enzyme substrate complex, and this can't, because of this structure is not fit in the active site. They both are different, so no reaction occur. But there the substrate structure and active site structure are same, so this is substrate attached with the active site of the enzyme and make an enzyme substrate complex. Further it make a product. Okay, now next one is a mechanism of the enzyme catalyst. Okay, enzyme is a catalyst that increase the rate of the metabolism. What is metabolism? In our body there is a growth, there is a excretion, digestion, respiration, all are called a metabolic reaction. So enzyme are large protein, and each type of enzyme has a specific structure to catalyze a particular biological reaction. Means the enzyme is a specific, that is the features of the enzyme. Now chemical reaction. So in the chemical reaction there is a two type of point: activation energy and transition stage. So the reaction molecule must gain minimum amount of energy, that is called activation energy, and to initiate a reaction and get activated stage, which is also called a transition stage, then form the product. This energy of activation can be lower by increasing the temperature of the reaction medium. But in the biological reaction, the energy of the activation is lower by the use of enzyme. Okay, so there is a three step of the enzyme catalyzed reaction are: first is a binding of the substrate. So enzyme plus substrate make a enzyme substrate complex. Second is a conversion of binding substrate to the bounding product, मतलब it make a product. Okay, last one is a releasing of the product. So product can get released and enzyme get back their original shape and position. So this is a graphical representation of activation energy of uncatalyzed and catalyzed reaction. So this is transaction stage. And what is a transaction stage? To initiate a reaction and get activated state, which is called the transition state. This is uncatalyzed stage. This is catalyzed stage, means in this position enzyme is not present, but in there enzyme is present, and this median line is called the activation energy, means the reaction molecule must gain minimum amount of energy, is called the activation energy. Okay, and this is the free energy when product get found. So enzyme dissociated with the product and release energy. So this is the free energy. This is a graph, x axis and y axis. This is a free energy and this is rate of reaction. So this is all about the graphical representation of activation energy. Next, move to the binding substrate theory. Binding substrate theory means you can say that enzyme bind with substrate theory. How can the enzyme bind with substrate? There's every scientist have a different opinion. So there's a different theory, but two theories are very important: lock and key theory and induced fit theory. So let me talk about the lock and key theory. So this theory introduced by the M.L. Fisher in 1980. Okay, and this theory, this is the picture description of this theory. Okay, so in this theory we can say that the specific key can open only a specific lock, as the name is shows. So in the same manner, the specific enzyme can transfer only one specific substance in the product, means specific enzyme can bind with the specific product and make a complex. Okay, so according this model, the active site has a rigid structure. Rigid means hard structure. It cannot be modified. It cannot be small and bigger and modified into different structures. It have to be rigid, hard structure. There is no modification flexibility in the active site before, during and after the product forming. Okay, so this is the lock and key theory. In this theory, the enzyme have a the enzyme active site has a rigid structure. Second is the induced fit theory. It is also called hand and gloves theory. Hand and gloves theory, and this proposed by the Koshland in 1963. So what is hand and gloves theory? So when we wearing a glove, so gloves can modify their shape according to our hand, and they can fix in the hand. They didn't move. Okay, there is a flexible structure. They don't have rigid structure. So in this theory, they propose that the enzyme molecule are a inactive form. To become active, they must undergo a slight change in the structure to move specific to according a substrate. So the active site can be slightly modified their structure. So this is the enzyme and this is substrate. This is structure and this is structure is different, but there this structure is same. So they can form a product, but there is a slight change. So the enzyme get modified their structure according to the substrate, so that the substrate get bind in this structure and make a enzyme substrate complex. Okay, means enzyme change their shape slightly to fit, and wait a second, enzyme change their shape, its shape slightly to fit substrate. Okay, substrate. Okay, so there is a two main important theory: lock and key theory and induced fit theory. And now other mechanism are catalysis by proximity, means there is the reactant must come together and bind with enzyme in proper orientation for catalysis to occur. So there the rate of reaction directly proportional to the concentration of the reactant. Second is the acid and base catalysis. So their organic reaction are catalyzed by acid and base, means specific acid and base catalysis there is a, and general acid and base catalysis. In specific acid and base catalysis, in this the rate of reaction catalyzed by the hydrogen and hydroxyl ion, and in general acid and base catalysis, their rate of reaction is directly proportional to the concentration of the acid as the catalysis of the proximity. And third one is a covalent catalysis. Covalent, there is a covalent bond form. So rate of enzyme reaction may be enhanced by the form of the covalent bond between the enzyme and substrate to produce the covalent intermediate, highly reactive covalent intermediate. Okay, covalent bond, I told you, there are two atoms, they are sharing their ions between together. Okay, so next one is a substrate strain, means substrate and enzyme are held together by binding forces. So this force causes strain the substrate molecule so that the substrate can distorted, means twist its shape during the enzyme substrate binding. So this substrate strain just opposite to the induced fit hypothesis. Induced fit hypothesis, in this the enzyme get modified according to the substrate, but substrate strain there is a enzyme put force on the substrate, so that substrate get changed and modified according to the enzyme, so that they can bind and make a enzyme substrate complex. So finally enzyme and substrate have to bind and make a complex, so that the reaction going to be activated and metabolism going to be activated, otherwise there is no option. So this is all about this. So enzyme activated site, there is the active site, how they attach, features of active site, mechanism. So there is the two chemical reactions, then binding substrate theory, this is very important, lock-in key theory and induce theory. You can write down this figure also in the examination, and last these four other mechanism. Okay, so thank you for watching.